Hi everyone. In today's video, I'm going to be going through the structure of a lymph node, as well as exploring the mechanism by which lymph, the fluid, flows through that lymph node. Now, the image that I've got on the slide here is actually quite a simple image. There are many other different parts to a lymph node, but these are probably the key parts that you would be expected to know regardless. So I'm going to start off with this image, and I may add a little bit to it, but that may be regarded as extra information depending on what subject you're learning and how your teacher does your subject. So let's firstly start off with the lymph node's structure. And the first aspect that I want to talk about is the outer core, or the outer covering structure that is present on a lymph node. So all lymph nodes are surrounded by a capsule, which is this part here. And the capsule is, in terms of connective tissue, made up of a dense fibrous capsule. And this dense fibrous capsule is collagen. Now, this capsule essentially allows for protection of the lymph node. So the interesting thing about the capsule that covers the lymph node is that, as you can see in the diagram here, not only does it allow for protection of the outer parts of the actual lymph node, but it extends into the lymph node structure itself. And what these extensions are called, and I don't think it's being given on the diagram here, but it is something that you should know, these structures are called trabeculae, or the singular is trabecula. Now, trabecula what divide the node into different compartments, and as you can see, these reddish areas here are divided in their structure by these trabeculae. So by dividing the trabeculae, by allowing for division, it allows for a certain amount of organization in the structure of the actual lymph node. And the lymph node itself, along with the divisions formed by the trabeculae, also contains a cortex and a medulla. Now, the cortex, as denoted by the picture here, is given by this range. So the cortex is essentially the part of the lymph node through which, or the extent to which, the trabecula extends into. And that makes sense because the cortex is also defined by the area in which you see the germinal centers. Now, the germinal centers, which are the red parts on the diagram here, are the parts of the lymph node where we find B cells. So whenever an infection occurs in the body, the microbe is able to enter the lymph node through the afferent lymphatic vessels, which are the vessels present at the top. And once it enters the lymphatic vessel and passes through the various tunnels and channels, it can enter the germinal center, which is where only B cells are present. And if a B cell that matches the antigen of the microbe comes in contact with that microbe, then it can proliferate and cause for an increase in the size of that germinal center. In fact, so much proliferation of that lymph node or that germinal center occurs due to the replication of the B cells that the lymph node itself increases in size. And therefore, that is the reason why, say for example, if you get a sore throat, your lymph nodes around your throat become inflamed as well. And that's why your doctor, if you go to your GP to say that you have a sore throat, and your GP might put their hands around your neck and feel for various aspects, they're simply feeling for if the lymph nodes around your neck have become swollen. And if they do sense a swollen lymph node, then that can indicate the presence of an infection. So the cortex, to reiterate, contains densely packed follicles with the germinal centers, and I'll just write down here. So they contain the germinal centers, which contain the B cells, and the T cells are located in the deeper regions of the node. And the deeper regions, which are pretty much this area here, is defined as the medulla. So the medulla is where the T cells are present, or what we call the T cell area. And it is where dendritic cells, which are special immune cells, which are having very large projections, are present as well. And the medullary cords are also present. So the medullary cords are interesting because they're essentially large streaks or cords, essentially, or wires that are present within the medulla like that. And these wires are so interlaced and interconnected with one another that they actually can allow for the trapping of various microbes that may be present within the lymphatic fluid. And therefore, by trapping these microbes, the T lymphocytes that are also present on these medullary cords can come in contact with the microbes in order to allow for an immune response to be incited. So that's another smart way in which the lymph node actually allows for protection of the body. So once again, I'll probably just draw them as a crisscross here. So these crisscrossing structures are referred to as medullary cords. Now, the last aspect that I want to talk about in this video is the pathway of lymph through a lymph node, and address a concern that may be present with some people in reference to the afferent and efferent in lymphatic vessels. So with lymph nodes, essentially lymph enters through the afferent vessels, which are present here, and that makes sense because afferent refers to A, or going in, and after, as denoted by these arrows, after the lymph fluid actually flows into the vessel, it enters an area called the subscapular sinus. And just looking at this image here, I don't think the subscapular sinus is actually present, but the subscapular sinus is essentially an area that is present between the actual cortex and the inner germinal centers of the lymph node, and that is where the lymphatic fluid enters first. So let's just say this area is denoted by the subscapular sinus. Alright, now from the subscapular sinus, which is, sorry, I've been saying subscapular, it should be subcapsular, because it's below the capsule. I'm really sorry about that. Subcapsular sinus then moves into the smaller sinuses. So this is where the lymph fluid essentially percolates from the surrounding cortex to the medulla. So the smaller sinuses are essentially these parts here, which are denoted by the borders formed by the trabeculae. So after moving through the smaller sinuses, through the cortex and into the medulla, the fluid percolates through now the medulla sinus. So the medulla sinus is this area once again, which I showed before. And I'll just quickly draw in what I've been talking about. So these are the smaller sinuses. This, in addition to the medulla, also contains the medullary sinus. And after moving through the medullary sinus, the lymph fluid exits the node at the hilum. So the hilum, as denoted in this diagram here, is this region. And essentially, it also contains the region in which the sort of capsule folds into itself at the borders at which the efferent lymphatic vessels are present. And it also contains the lymphatic veins and arteries, which are the veins and arteries that supply blood to the lymph node. And the hilum is essentially therefore a key area where the exchange of fluid occurs. So lymphatic fluid then flows through the efferent lymphatic vessels after passing through the hilum. Now, a key feature that you would have noticed due to the structure of the lymph node, is the discrepancy that we see in the number of afferent versus efferent lymphatic vessels. And unfortunately, this is where the diagram is a little bit inaccurate, because essentially there are more afferent lymphatic vessels present than efferent lymphatic vessels in a lymph node. And you may be wondering, well, why is that the case? The reason why this occurs is that, firstly, when lymph is flowing into a lymph node, it's flowing at quite a fast rate, since the entire job of a lymph vessel is to simply allow for the transportation. But the job of the lymphatic node is to allow for monitoring. So if we have a discrepancy between the afferent and efferent vessels, then if less efferent vessels are present and more afferent vessels are present, then lymph fluid is going to be slowed down within the lymphatic node. So if we were to represent the lymph node as a bucket here, and say for example we have 3 taps which are emptying water into this bucket, but we only have 1 hole at the bottom of the bucket. So as you can see, even if the flow of water into this bucket is going to be very high because there's 3 taps emptying into the bucket, since there's only 1 hole at the bottom of the bucket, the flow of fluid out of the bucket is going to be very low. And that's the same concept that is applied here with the lymph node. Since there's only 1 or maybe less openings compared to the inlets into the lymph node, there's going to be a slowing down of fluid and the ability of the fluid to actually be monitored. So that concludes this video on the lymph node. So today we explored the lymph node structure and how lymph actually flows through the node, as well as addressing the discrepancy that exists between the number of afferent vessels and the number of efferent vessels. So I hope it has all made sense today, and thank you very much for watching.